Greetings, this is Tien and Hao. Today we are presenting on biomarkers for responsiveness to Folfirinox chemotherapy combination for pancreatic cancer. According to Cancer Statistics in 2021, about 60,000 people were diagnosed with pancreatic cancer every year. Prognosis and survival rates of pancreatic cancer remain poor as we fail to detect it at early stage, and there are many complications associated with standard treatments. This study hypothesized that there are differences in gene expression in response and non-response groups in 5-fluorouracil plus flucovin plus irinotecan plus oxaliplatin, known as the Folfirinox combination. The findings from the study can help predict biomarkers that can predispose patients to current standard treatment responsiveness with standard chemotherapeutic agents. Our results should aid in better understanding of disease condition and any drug development to target these biomarkers.
So with the data from Pancreatic Cancer Network, we were able to identify 28 patients with gene expression data. 9 of them was responsive to Folfirinox, while 19 of them did not respond. This list of genes was gathered using the bioconductor package DSEC2 tool and ran through different analyzing tools, including cytoskip, which we look at the string protein-protein interaction, the reactant PA we use to identify pathways that are overrepresented in the genes, as well as we use the gene set enrichment analysis to identify gene sets that have statistical significance between responder versus non-responder. With the data, we also put them in machine learning to test for predictivity of responsiveness to standard chemotherapy.
With overrepresentation using reactant PA package, we were able to identify keratinization as the most significant biological pathway in patients who did not respond to treatment. To further investigate protein involvement in keratinization, we used cytoscape software where we were able to input 233 differentially expressed genes to generate a protein-protein interaction network that consisted of 138 proteins with 53 interactions, in which cross-linked envelope proteins of keratinocytes were found to be closely related to other protein-rich proteins, which is a major nutrient source for tumor metabolism, as well as proteins from other keratin families such as KRT or KRTAP.
So while the reactant PA look at individual genes, this tool helps us look at the gene set, which were, in this case, the GOBP keratinization and the reactant keratinization gene set that has statistically significant in the non-responder group. So from this analysis, we were able to identify a list of leading-edge genes, meaning those genes that hit the enrichment score, and with the nominal p-value of 0.001 and the full discovery rate of 0.001, the keratinization gene set once again was highly upregulated in the non-responder. And with the identified pathways as keratinization, we put them in machine learning using the AWS and the random forest scheme, using them to predict responsivity to Folfirinox. And as we could see, these two schemes, both with tenfold cross-validation, were able to identify or predict the non-responsiveness with a precision rate of 0.88 for AWS and 0.77 for random forest. Similarly, with the response rate of 0.78 for AWS, 0.89 for random forest, the F-measure rate 0.83 for AWS, and 0.82 for random forest. Please keep in mind with this result, we actually have a better predictivity of non-responder because there's actually more patients that did not respond to this combo than the responder, which make it a limitation.
So our main findings from our study include identification of keratinization and feasibility of employing machine learning in predicting responsivity of chemotherapy based on the overrepresented pathway. Our data capture keratinization as the most significant biological pathway in the Folfirinox non-response group, in which cross-linked envelope proteins of keratinocytes and proteins from keratin family were identified, although not shown to be associated with keratin 17, which was identified in literature, was significantly upregulated in cancer proliferation and chemoresistant in pancreatic cancer. Our machine learning algorithm was also able to predict up to 78.6 accuracy in tested populations using tenfold cross-validation.
In conclusion, the keratinization pathway identified from the differential expressions from the data make it a viable biomarker for precision treatment for pancreatic cancer and help with further investigation in developing new therapies targeting at this pathway. The other pathways overrepresented in non-responder can also be studied using biomolecular techniques and adding this to the machine learning model in order to make them a more comprehensive tool in identify responsiveness versus non-responsiveness to chemotherapy.